Hello, everybody. Today we're going to be talking about Claude Science. And I am Sanker Jr., Director of AI and Business Development over here at Bay State IT. Been helping lots of different biotech companies with their AI strategy, their implementation, building agents, executing securely, etc. We have been getting a lot of inbound requests on Claude Science specifically, pretty much actually immediately after it was rolled out. I had gotten into the actual initial presentation where they announced Claude Science, and I think maybe an hour or two after it, I was getting emails, we were getting tickets, and we essentially said, you know what, emergency webinar. We did it 2 weeks out so I could do a little bit of an investigation, see what it actually offers, see what it does internally, the risks, etc. And this is the culmination of that. So today we're going to be talking about Claude Science, how you can use it, some of the positive parts of it, why it's useful, why it's helpful, what to watch for in the public beta, and then also how to roll it out securely because, well, it is in beta. And it's not production grade software yet. They're expanding into science, which is incredibly exciting. We're very happy to see that Anthropic is investing in science, as is OpenAI with Rosalind and other things like that, though they are less less willing to immediately roll these things out to people. I digress. So, what is Claude Science? Well, as I said, it's essentially Claude code in a trench coat. But what that really means is it's kind of a workbench. So Claude Code is a agentic software engineering platform system. You go into Claude Code, you talk to it, it runs commands, it talks to you, you work with your data, it's able to browse and use your computer with you, write code, execute that code. This takes all of that, puts it into a more friendly UI, and then it also adds in some pre-baked connectors, skills, and user interface elements that make it easier to interact with this system. So it is an integrated AI workbench, and it takes a lot of those different tools that might be more difficult, say, to on your own go out, find the tools, configure them correctly, understand what's helpful, put them in the right place, and then use them in one interface. It does all of that for you. So for a lot of people, they're using it for literature. It's already got built-in integrations with a few of these data sources here that are listed out on the slide. It's fantastic for working with lab data. You put the data into your folder, you're able to then open up Claude Science. Hey, take a look at my data, run some analyses, get some initial statistics, metrics, walk me through my data. What can we do with this? Here's something I want to do. How can we do this? Right? And then take that information and build a workflow into it, build agents and skills, which we have another webinar coming up about what are skills, what are agents. Everyone's talking about building agents, and right now, most people are talking about building skills when they're talking about building agents, but we'll get into that in the next webinar. For today, you can just know that you can use Claude Science to go through some process, to understand it, to get some evals, some metrics, some validation data, and then to pack that into something reproducible, which is pretty exciting, especially if you can share it with your coworkers. So under the hood, it comes pre-baked with its own agents. And as you recall from what I was just saying with agents, these are really more or less skill systems, but they're still incredibly helpful. So we've got a generalist coordinator, which is what you're talking to most of the time. They have some domain specialists for genomics, single cell, proteomics, structural biology, cheminformatics, a reviewer agent, which is very interesting. Personally, I've found that having an agent with a fresh context window review work that's been completed is incredibly helpful. When you ask an agent that's performed the work itself, hey, how did you do? Well, usually when someone does work, they're going to think they did a good job by default. If you clear the context window, which is what they have pre-baked. So a lot of Claude Science is pre-baked workflows for you that already work fairly well. This is something you could have made yourself, the reviewer agent, but this makes it much easier to use. So the reviewer agent, fresh context window, goes through the system and will go and review the work that's been done, look at the data, flag issues, flag assumptions, and then ensure that everything lines up. And we've got some partner models here, which is very exciting. I'll show that a little bit on the demo that we actually walk through. So we can, with this tool, get more reproducible and auditable results, specifically because you can see all of the code. When you run through these computations and these processes, you're actually going to be getting these snippets as it goes along. And you can even click Notebook on the bottom bar right above the chat window, and that'll pop open what looks like a Jupyter notebook, which is pretty neat. I think it's peeking into essentially the IPython runtime environment that the agent offers. I'm not going to dive too deep into it. I would love to. It's lots of fun, but it's not necessarily, we don't have enough time, and it's a little in the weeds there. But what this means is you can see what's been executed in a format that just looks like a Jupyter notebook that you can take yourself and bring into Jupyter. You can bring it into Colab, anything that you want, work with it there. So that's been really exciting because a lot of people, they've worked primarily with the web interfaces, which are great, don't get me wrong, but the web interfaces don't make it easy to see and understand. I mean like Claude.ai or ChatGPT.com. At least historically, they haven't made it easy to see and understand the processes that are actually occurring, the code that's being executed, and to be able to take that without, say, explicitly asking the model. Claude Science brings that to the foreground and makes that a main feature of the system because it knows who's targeted here. Scientists that want to be able to have reproducible work. When you're running this code, it's all running on device. So only the context is sent to Claude. Your chat window, absolutely. That's sent to Anthropic. Now you have to understand the data agreements, and we'll get there later, but this is the same sort of risk as any data that you're inserting into an AI model. So long as you don't insert necessarily the raw data itself, that's not being uploaded like some companies that maybe some people have seen some news articles about that have been uploading entire code bases to their cloud. I digress. This is not doing that. If anyone was curious what I'm referring to, it was shown that Grok's CLI from Elon and Twitter was uploading entire code bases to their remote system. I would recommend don't use Grok, but I digress. That's actually in a different webinar where we talked about what AI models to choose, what platforms to choose. But this one, your data is not aggressively sent up to Anthropic. There are things to consider, and we can talk about them. What is your acceptable use policy? What's data is allowed? But by default, you're just going to be sending metadata. Say, for example, you ask Claude to run some statistics, some information, provide me some metrics on this data. When it outputs that and it checks it, that goes up, but the raw data does not inherently go up. And then something that's really cool about this is you can actually connect to your clusters with this. You can give it remote access, you can give it AWS access, SSH access. It can go into remote ecosystems and execute code and give you results, which means something that you may or may not be able to run on your laptop, you can then offload that work remotely. It's very cool stuff. So why is this helpful? Well, if you have one place to do everything, that makes it a lot easier. Software engineering and computational bioinformatics is complicated. It's difficult, and this tool makes it easier to get started, and it makes it easier for other people to get onboarded in your team, to share workflows, share skills. Whereas before this, you would use something like Claude Code, which again, I've said many a time is a great tool. It's wonderful. I love using Claude Code, but it's also a bit advanced for most people. And this makes that easier to work with, easier to share, and I hope Anthropic will continue to invest in Claude Science and science in general, and will make this even more advanced and a better tool for you to use with your team. Frankly, right now, it's pretty good. It's also great for making things faster. If you have one place to do everything, you can share these workflows. There's less startup time. And a lot of the time, when you're trying to get someone onboarded, I don't know if anyone's ever emailed you a Python script and is like, hey, run this script against the data. It's awesome. I spent 2 weeks working on this, and this is my analysis.py, or just a Jupyter notebook. Fantastic. You open it, you run it. Could not find module pandas. Oh, whoa, okay, pip install pandas, no problem. Conda install pandas. Data frame dot interpolate does not exist. Wait, what do you mean? Dependency issues, pain in the neck. You can work with Claude. Hey, here's this thing. How do I get started? how do I run this? Claude will work with you to get that started. So I would say actually working with Claude Science to have it walk you through and help you figure out how to do computation is a really valuable skill and a valuable tool. As we have here, that scaling, jobs landing on the right hardware, though I would recommend you chat with it, have Claude interview you. Hey, where do you want this to go? We can discuss that more later. Skills are fantastic. And then this institutional knowledge, this is really important and something that we're going to talk about again in the future with more skills, but it is useful and you can share skills among your enterprise if you have that configuration set up to be able to share among your team. So this is a niche, research-focused product. It's not going to be for everyone. It's not going to be for every department or every role, but for those who do benefit, they will benefit a lot. Some results from some beta users that we've been seeing in the ecosystem. Well, this is specifically from the launch announcement. Manifold Bio using Claude Science to identify and prioritize drug candidates. It scored each candidate against the company's own proprietary criteria. Allen Institute, a single comprehensive literature review used to take a team up to 2 years. They've now produced around 10 of them, many over 100 pages long, with a reviewer agent checking every citation for accuracy along the way. I just want to pause there for a second. Key detail, reviewer agent. Why? Why do we do a reviewer agent? Because you shouldn't assume by default that output from AI is accurate. And so this is a very important part of it, and this is baked into Claude Science, which is pretty neat. And then the UCSF Brain Tumor Center completed germline genetic workups, comparing several analytical approaches side by side in what the team describes as roughly 1/10 of the time the same analysis used to require. Figures above are self-reported by the teams and not independently verified. The through line here being, it helps reduce time-draining tasks, and it makes something that might be difficult for someone to pick up on their own easier to pick up because Claude can walk you through it. So, shall we see what Claude looks like? Claude Science. I asked Claude, hey, what are some cool things that we can do with Claude Science that would be a fun demo? You can see here, what are some pretty examples I can run for a webinar demo I'm doing of Claude Science later today? So, let's pop in. And sure, something that is a good place to start here would be, how about caffeine? I mean, I was just drinking some coffee. One moment. All right. So build the structure of caffeine and open it in the Catcher Sketcher so I can edit it live. Then compute and show me its key physiochemical properties, molecular weight, you can see the rest there, and tell me whether it passes Lipinski's rule of 5. Interesting. Good prompt. So, what's going on? This is a deceptively complicated and simple request. I want to just stop for a moment and read the chat log because we're not doing this live. We're doing this pre-baked, but this will give us a little bit of an idea of what it's actually doing. You know what? I'm going to go broad level first, then we'll dive a bit deeper. So you can see I'll build the caffeine, open it in the sketcher. I'll create a dedicated environment. Let me build it. Let me save the structure. This to failed is interesting. We'll get back to that in a second. We're going to do broad overview, then we're going to dive in. Mol artifact is saved and will render. I wasn't able to auto launch it programmatically, but here's the full property file. So here's the structure and the physiochemical properties. We've got the molecular weight, pass, yes. I believe that's the reviewer passing there, 0 violations. Log P, and we've got a few other stats here. It satisfies all 4 Lipinski criteria comfortably, with substantial margin on every one. So you got some analysis, and then we have a saved artifact of caffeine. That's cool and all. We're going to have to go open this in PyMol, right? No. Actually, you can open this right in the browser. It's going to take a second. Cue the elevator music. And here we are. So that is it rendered. How neat is that? And then we can draw. Now, the last thing I know how to do is draw a molecule, but I can show you how neat it is that we can do that within here and that it comes pre-baked into the system. So pretty neat. We've got a general chat window, and then we've got some results. I'd like to dive in a little bit to what it's actually doing what makes this powerful and what makes it a little bit dangerous at the same time, which is why we recommend pausing. We'll talk about that later. So what's happening here? We're going to check for an RDKit environment. First, list dependencies, RDKit. Well, here's Python. We're missing this. Looks like we do have these. R is installed, but we're missing RDKit. Check 2, no environment has all of them, showing 2 closest. Well, it's going to go ahead and create an environment. That seems sweet, right? Yes, yes and no. It's installing RDKit live from CondaForge autonomously on your machine without you interacting with it, which is incredibly powerful and potentially a little bit dangerous. You can see here, let me build caffeine and generate the mol file. So we've got the smile string here. We're going to go and create it from the smiles, write the block, and then it's going to print out that info that it's running through RDKit. And you can see all the various functions that they're running here. And you can see the output. That's actually Python code that's being executed live on your computer. What was really interesting here was where it was trying to save it. We were able to go ahead and save that file, and then it failed to open. Kernel subprocess terminated unexpectedly because Xcode select is not installed on my VM. But despite that fact, it actually was able to figure it out itself. So it can run into failures, run into issues, and then recover from it, which I think is very cool. And as I said with the notebook feature, discard changes, you can actually see this as a notebook. So that's, I think, the most complicated one I did. I'm going to go, not the one I just did, but the one I just clicked on. I'll go to that after. And we can't do all of these demos, so I'll just do maybe 1 or 2 more. And so we have another one here. We are fetching the crystal structure of deoxyhemoglobin, and we're downloading it, file downloaded. It says actually it's oxyhemoglobin. Again, not a biologist. But let me confirm. Confirmed. It's this, which is pretty cool. So I actually said something wrong in my initial query, and it corrected itself after getting data. One correction up front, it's going to call it out, it's going to explain it, and then show what you're looking at. And here is this, here is that. And to highlight it live, the cleanest single residues to grab is this one. Okay, well, let's go ahead and see. Oh, would you look at that? I don't know if it's conveying well over the screen share, but we are live rendering that PDB file in a 3D mol.js viewer, which is incredibly cool. Now, if I, someone who is not a biologist or chemist, can do this without much knowledge, imagine what you could do, where you do know exactly what you want to do and how to do it. So that's pretty neat. And again, we can click Notebook here, and you can see the actual code that was executed. I'm going to do one more demo here, which is a little bit more complicated. And then I'll talk a little bit about the reviewer as well. But I'll let you read through this if you'd like. I'll pause for a moment on the task, assuming that you all can read it. How long would it take everyone to run this? If you had this task, you were assigned it at work, and someone asked you, complete this, what would you give as a deadline for how long this would take to execute? I would say, personally, this would take me a moment. So how long did this actually take Claude Science to do? I think it took 15 minutes with 0 feedback from me. So I will not go through every single cell, but it's pretty interesting. First and foremost, it actually drafted out a plan. Because this is kind of complicated, it said, well, here's my plan. What do you think? It proposed this to me, load the data, do the filtering, normalize, PCA, neighbors, UMAP, dimensionality reduction, marker-based cluster annotation, publication UMAP by cell type, marker dot plot, and then save it. How's this sound? It proposed to me, and then I clicked approve, and it said plan is approved. I could have provided feedback if I wanted. It's gone ahead and set up the environment, and I'll go ahead and open the notebook on the side so you can see maybe a notebook view of this. This one's a lot more involved. So all of this here, it's running through, it's fetching data, it's running the processes, it's doing the QC metrics and filtering of the cells. You can click on these to see them. It's running normalization, log transform, PCA computed, only the plotting cell rejected X. Let me plot it without X and continue. One of the through lines here, actually that seems, I don't know about y'all, but that seems like it might be missing the line. Or maybe I just do not know this specific graph. But proceeding here, and then you can see the clusters have been formed, which is very exciting. And later on, it's actually gone ahead and it's labeled those clusters, which is super cool. And then it read the image itself, because this is a multimodal model. It can read images and provide assessments. So it says, oops, these labels overlap. Let me go ahead and do direct labels and leader line labels so that they're not overlapping, which I think is very cool. And then it's corrected something again. And it's going to create a marker dot plot. And here's its results. It's going to provide all that information, publication UMAP by cell type, marker gene dot plot, cluster to cell type assignments, and its deliverables. And then here you've got the entire notebook. Should you want to go through, copy the code, run it yourself, take this, turn it into a Python library, make it something that can run on your servers automatically based upon some data ingested into an S3 bucket and your Nextflow pipeline, anything you'd like to do, you can do with this, which is pretty neat. And that is most of what I wanted to cover of just walking through the general interface. As usual, we've got some standard stuff here. The reviewer, I'm actually not seeing it immediately popping up. It was popping up when I was running it, but that's pretty interesting. So auto review seems to be disabled right now. Background review when work completes has review. Actually, I don't want to ask in there because that would take forever. Has a review been run? Can we run one? And you can also see the reviewer model that you can select, whether or not memory is enabled. There's specialists, which I'm not going to talk about right now. They're a little bit more in the weeds, but they're essentially just pre-configured agents. Specialist is a now overloaded way to say agents, but no review has been run. What kind of review would you like me to run? Let's just go ahead and it's got pros and cons for each, which is great. Let's just do the simple one, whether or not the numbers are correct. And you can also see the compute destination, whether or not you want to do some cloud compute or an SSH host that you want to delegate to. That's in the weeds for this, so we're not going to go into it. This is just meant to be sort of high level. And while it's running that, I'm going to go ahead and pull up some of the settings. And you can see here's all the skills that it comes pre-baked with by default, which is pretty cool. It's got skills for AlphaFold. It's got skills for bolts, Borzol, Chai one, is that Chai? Font small. Yeah, Chai. A bunch of different little snippets of information so that it can do these specific tasks very well. And then it's also got some pre-baked connectors, 24 by default. When you're setting up Claude Science, it'll ask you if you want to enable all of these, disable all of these. You can go and enable and disable on your own as well. You can configure specialists, which are very interesting. They already have a reviewer specialist. You can create your own specialist either by chatting with Claude or writing it from scratch. So you can see how you could fill that in. It's got instructions, capabilities, very cool. Memory, compute. Memory is ways to have it remember things about you. Compute would be where you're setting up your third party, well, your remote sources of processing. So you could do an SSH host, which is pretty interesting. You can also do a cloud provider like Modal. Pretty neat. And then network. This is fantastic. It sets up the ability to filter what kinds of network requests can be accessed. You see here, I allowed Alex Wolf's data store because it was having trouble downloading the data sets for the cell clusters initially. And you can also see here, very interesting, some of the usage. I'm sure everyone's once or twice run out of usage with Claude. So here you can see the usage, and they've even added some really great stuff around where it's happening from. We've got caffeine, it's occurring there, it's occurring here as well, and then details on specifically assistant pros, tool calls, and artifact provenance. Pretty neat. And let's see. Are we done here? So it is still running the review. You want a QC review, it's doing that. Wow, looks like maybe it found one discrepancy, which is why the reviewer is so important. And this is also beta software. And like we say in all of our AI trainings, never rely on AI deterministically to provide, or never rely on AI to have the correct results every time. Always verify you are responsible for your own work. AI is not going to take responsibility for something going wrong. And that's why they have a reviewer. It's an interesting byproduct of these kinds of tools that you can't assume that it's accurate. And so even a review isn't going to guarantee that it's accurate, but it adds one more layer of accuracy, or at least improving the accuracy. Okay, and it's done. No review had run, so I ran one. Well, the result values are correct. These are the results that are correct. 5 of 7 match correctly. The two that differ are not errors. They trace to legitimate algorithm choices. So, different algorithms produce different stats. Pretty interesting. The discrepancies don't touch any Lipinski criterion. Cool. All right, so this is in public beta. It is not a production grade product yet. They're actively developing it. They're working on it. It's very exciting. But it's early access. It's only on Mac and Linux, no Windows at the moment, and it's a local agent that can run code, manage environments, and reach your files. That's very useful, but it's potentially dangerous. Why? Well, because it can read your data, access your files, and execute arbitrary code and install packages on its own. We did a review of its behavior and its executions. We currently consider it a medium high to high risk product. A, because it's beta, B, because it's a local coding agent on your device. Great power comes great responsibility. What can it do on your device? It can run code, it can install packages, access local folders, store credentials, install packages, and update itself. So it can update its own system. Very interestingly enough, it actually seems to just run through a web browser, which is pretty interesting, but this is not malicious. It is just what it needs to do to work. It's inherently part of its functionality, but it's risky. And the other thing here is, well, data and regulatory exposure is something you need to think about. You need to check with your lawyers first to make sure that this is information that you're actually okay to be transmitting and that something doesn't need to be sorted out first, like an AI policy, acceptable use, permitted data, which if you don't already have, you probably should have, and we can help you with. IP. If you're worried about this data going out to Anthropic, Anthropic could get hacked. Hopefully not. It's not occurred thus far. But anyone can get hacked, and you just need to accept what's your acceptable risk, what data is allowed, what data is not allowed. And then one big risk here, I don't know if anyone's heard about the big NPM security incidents, the PyPI, backdoors, worms. Well, open source software engineering is phenomenal. Open source has made so much of our ecosystem what it is, and it's helped the world grow in substantial and meaningful ways. It's also relied a lot on trust on third-party developers across the ecosystem to be publishing good code, and most people do. You check, does it have a lot of stars, lots of downloads? Does it seem to be okay? You install packages, you update them periodically. Well, recently, that trust had some issues because a lot of packages had been compromised. Malware was distributed, info stealers, and specifically with Shai Hulud, there were worms, so self-propagating malware that was expanding across the ecosystem. We recommend for your package managers, bare minimum setting a 7-day cooldown cycle for installing packages, which means never installing packages that are newer than 7 days unless you explicitly permit them to be installed by permitting and omitting that singular package from the policy. Say, for example, because there's a 0-day vulnerability that you must patch in your application. Other than that, we recommend you wait at least 7 days because usually virus scanners, AI virus scanners, AI that actually checks for malware, static analyzers, usually catches this kind of stuff within sometimes an hour, max 2 days, usually not max as a hard rule, but usually is caught within 2 days, so 7 days is a good minimum. Unfortunately, right now, Claude Science does not have any of that pre-configured. That may have changed since we last analyzed it, but when we had done an initial investigation, it was installing the latest version of packages as soon as it went to install. So you saw it installed RDKit there. That's awesome. That's super helpful. But if RDKit got compromised 6 minutes ago and it auto-installed RDKit, you now have malware on your device. You don't want that. So this is why we suggest extreme caution and care to those who are using it. How do you use it securely? Well, first and foremost, if you're on Claude team, unless your entire team should be permitted to use Claude Science and is trained and aware of how to use it properly, we recommend not enabling it. Why? Only Claude Enterprise allows group granularity and access control, where you can, say, have a set of early adopters or a set of trained individuals. Early adopters, same thing, right? People who are aware of the risks, have been trained, say, have seen something like this, and we actually have more advice than, well, we've offered in just this webinar for how to use it securely. I've kind of just given a tour. I haven't given that many tips, but individuals should be trained. They should be aware of what can and cannot be put inside of Claude Science, how to use it, and if you're not going to do that, I would not recommend rolling this out yet. So the general bottom line being, Claude Science should not be enabled for broad employee use today. Adopt it deliberately and scope pilots with governance in place. And guardrails to put in place first would be access controlled only by a named group, data governance to make sure you understand what data can go in, compliance review of that, and then to treat it as an agent with arbitrary access to your file system and to be able to run commands. If that's not a risk you're willing to take, then don't install the product. It's as simple as that. What to tell your team? Well, communicate clearly who's in the pilot, who isn't, who's approved. Instruct users not to install it on company devices if they're not approved. And if they are approved, warn them that it's in beta. Expect instability and don't rely on it for production critical work. Keep sensitive data on-prem. Don't offload things to data sources and to third-party providers you don't trust. If you're specifically worried about those connectors, because connectors that come pre-baked and installed can upload your data remotely, don't enable the connectors. There are ways that you can mitigate risk or reduce risk with certain access controls and configurations that are available right now. So, to recap, Claude Science unifies literature, lab data, and writing into one auditable, reproducible workbench with a team of specialist agents behind it. It can accelerate drug discovery and cut the hours your team loses to wrangling data, but it's in beta. It's a local agent, and there's inherent risk. Hold off until it leaves beta or adopt it with scoped governance. Scope pilots and governance. We will be having a webinar on the 5th of August about building an AI agent with Claude. What are agents? Why is everyone talking about them? What do they actually do? So that will be lots of fun. We hope to see you there. And you can book a discovery call if you'd like to roll out Claude Science right now and you want to do it without risking, while mitigating the risk of data loss or security incidents. Reach out to us. We'd be happy to help. And you can stay connected here through both of those QR codes. So take a look there for booking a discovery call. We can help you with your AI implementation, rollout, building agents, training individuals, lots of good stuff there. Or just join us for the next webinar, or both. And I think we have time here for Q&A. If anyone would like to ask any questions, we got a few minutes. Claude Code, also very cool, very powerful. If your team is already using Claude Code, Claude Science is very similar. So the gap between Claude Code to Claude Science in terms of rollout and information is going to be less than a gap from never using Claude Code at all to Claude Science. So that's just one tip and bit of advice there. If you're already using Claude Code, your jump to this product is a less wide jump. Well, thank you all for joining. It was great having you, and I hope you all have a wonderful day.